This video is going to be an introduction to translation, which is step 2 of protein synthesis, where we take mRNA and turn that into proteins. Translation, as mentioned, is the synthesis of proteins from our mRNA message. It occurs in the cytoplasm and, of course, begins when mRNA leaves the nucleus and migrates to a ribosome, and we'll talk about that in just a little bit. And here are sort of the big players in translation. We have mRNA, tRNA, ribosomes, which include rRNA. So all three of our RNAs are involved in translation. And then, of course, amino acids, the monomers for proteins. And let's just talk about reading that mRNA transcript. So our mRNA is a complement to whatever gene was read during transcription. And we have divided, or we can divide, our mRNA into sequences of 3 bases at a time called a codon. So codons code for amino acids, and these codons make up what we call the genetic code. And this, of course, reminds me of, those little decoder things that sort of used to be old toys that you see in like A Christmas Story or whatever that movie is. And so the genetic code that you should know is basically universal for all organisms, which suggests that it evolved at one point a long, long time ago and has since been maintained in all organisms that exist today on Earth. And so if given this particular mRNA strand, we have divided it into codons. So with 3 nucleotides per codon. So there are 64 possible codons. How did we get that number? Well, if you think that there are sort of 3 spots, right? One for each nucleotide in a codon, there are 4 possible options for that first spot, right? A, U, G, or C. 4 possible options for the second spot, A, U, G, C. And of course, in a similar way, 4 possible options for that third spot. We end up getting 64 possible options. Okay, so there are 64 possible codons, but only 24 amino acids. Okay, and so this means that actually multiple codons will code for a single amino acid. So for example here, there are actually 4 codons that will code for arginine right here. You can have CGU, CGA, CGG, or CGC. All of those code for arginine. And I'm going to tell you how to read this wheel in a little bit, but you can see there are plenty of examples where one amino acid is coded for by multiple codons, and we call this the genetic code being redundant. Okay? A couple of different codons will code for the same amino acid. This is just another way to read the genetic code. So actually, I'll go back and tell you how to read this, I guess. In this wheel, for the codon, you actually start in the middle. The middle is your first letter of your codon. I'll do that in black. First letter, second letter, and then third letters. So if I have the codon AUG, I start here A, U, G, and that's methionine, which is actually our start codon. Another way to read the genetic code, and it's the exact same code, it's just read a different way. You can have your first base here. So if I again do AUG, I find A here. So now I'm looking at this whole row. My second base is the column, so I'm now looking at U, so that isolates that box. And my third base is here, G, that then finds me here, AUG, methionine, which is the start codon. So 2 different ways to read that. And so that's how we can go from DNA transcribed into mRNA, which we know how to do, just writing out the complementary nucleotide, divide that into codons, and then we get amino acids, which makes our protein. Here's just another way to visualize that. Here's our coding strand of DNA. Transcription happens and we get our mRNA. Divide that into codons, And then we get various polypeptides.